Good afternoon and welcome. Thanks for being here. I'm Lynn Katz. I'm a professor in the Environmental and Water Resources Engineering group, and really excited to have all of you come and to the 4th annual Lawler lecture. So, at some point we're going to have quizzes on who came on the first 3. If you saw the slides going through, you might get that. But this lecture brings together our community, our EWRE community, brings together former students. How many people here are former students? Awesome. Our colleagues in the professional world in Austin and beyond. Nice. And current colleagues at UT. Awesome. And also our new students. How many new students? Awesome. Thank you for being here. You can see this is our community, and I hope everybody has a chance afterward to meet and talk and get to know people of different generations that are here. I wanted to say something a little bit, especially to our new students who may not have met Des Lawler yet. And I wanted to say this. Des Lawler has been an institution at our institution. He was a faculty member here for over 40 years and really led the EWO program both spiritually and intellectually. He's an incredible mentor to students and faculty alike, and really served the students. And part of this lectureship really funds activities for the students, including the EWRE party and this lecture. To those of you who've donated, thank you so much for supporting this community. It means so much to us to be together on a day like this. In addition, I wanted to say something about one of the biggest contributions I believe Des also made, not just in terms of the graduate students he worked with and mentored, and what a great colleague he has been both to the people here, but also to the broader Austin community in terms of people who are in water and wastewater and even air for that matter. But one of his greatest accomplishments has been his textbook. And those of you who are new students, or probably many of you, are using this textbook right now. That was written by Mark Benjamin and Des Lawler. Not only is it an incredibly clearly written textbook that is very readable, gets through very tough concepts in a way that allows students to be able to engage with the book, but it's a great reference. And true story, yesterday, I actually used this book as a reference. I was looking for pressure changes across an electrodialysis membrane. I opened it up to the very last chapter, figure 1552, where there is a plot of potential versus distance across an electrodialysis membrane. It's a great resource. It's pretty exciting to be able to say, this is a really usable book, and this is one of the most significant contributions of the many that Des has brought to our field. With that, I want to introduce Des Lawler and allow him to have a chance to welcome you, and then we will move on to our lecture today, which will be from Dr. Raskin from the University of Michigan. Go Blue. Hook 'em horns too. But very excited to have both of them here today.
Okay, so I have a red face from all those nice things that Lynn said. Thank you very much, Lynn. I mostly wanted to thank all the companies and individual donors that was, on the slides coming forth. There's a tremendous number of people contributing to this, including, of course, my wife and I. Alice is here. But anyway, thank you for those donations. It really makes this happen, and hopefully this continues to grow as a great event in the EWRE outreach to the community as well as within EWRE as well. So thank you all for that. So I'm also wanting to take a poll, How many people here have been to all 4 of the Lawler lectures? Anybody? Oh, look at that crowd. Good job, okay? I have to, those of you who raised your hands to that might remember that I myself missed the first one. So, after months of planning and getting everything together and stuff like that, the day before, my wife and I both came down with COVID. And that was that. I mean, you just didn't show up. So there we go. So I've only been to 3. How many have been to 3, but not 4? Oh, another good crowd. Well, thank you so much. This is great. So hopefully, 10 years from now, we can ask how many have been to all 14, and there'll be a bunch of hands go up. So this is an annual event. It'll generally happen, about this time of year every year, and I hope you keep in mind. And if you didn't get emails about this and you want to be on a list, give me a card or a piece of paper or whatever with your email on it afterwards, and I'll be happy to make sure you learn about the ones in the future. So that's really all I wanted to say, but big thanks to all of you for coming, and big thanks to all of you who have donated. And so we have a lot of Michigan connections in our group. Lynn Katz, who just said go blue, did her PhD there. And I want Professor Xavi Finoli, who just joined us in the last couple of years, who did his postdoc with Dr. Raskin. I asked him to introduce her. So I know your program says Dr. Kiersitz will do that. Dr. Kiersitz's mother is in a hospital and quite ill, and she couldn't do it. Turns out she also did her postdoc under Dr. Raskin, which is why we asked her in the first place. So anyway, Xavi, it's yours. Thank you.
Well, hello everyone, and thank you very much for coming. I have the pleasure to introduce the speaker today. Professor Lutgarde Raskin is a professor at the University of Michigan. She got her bachelor's and master's degrees in bioscience engineering and economics from the University of KU Leuven in Belgium. After that, she moved to the US and she did her PhD in environmental engineering at the University of Illinois in Urbana-Champaign. She was so successful at her PhD that she got a faculty position at the same university right after that. And in 2005, she moved to the University of Michigan, where she's now. During her work as a professor, she has done research by applying microbial ecology concepts in engineered systems, especially systems for drinking water and also for wastewater treatment. She has worked with her research group in developing new anaerobic treatment processes by harnessing the power of microorganisms. And you are going to see this in this presentation. She's also she has also been the advisor for about 20 postdocs, one of them doing the introduction today, and about 100 graduate students. She's a fellow for the International Water Association, also the American Academy of Microbiology, and also for the Water Environment Federation and the Association of Environmental Engineering and Science Professors. In 2021, she also became elected as a member of the US National Academy of Engineering. And everything I just said, you can just read it here, which is really what I'm doing, right? So maybe I also going to give, I'm gonna tell you how I met Lut Raskin. That was the first time I attended an international conference in 2013 on anaerobic digestion. And at that time, I was a second-year PhD student, and I saw her keynote. And after seeing her keynote, that presentation was so good that I said, I really need to work for part of my PhD with this woman. This presentation was amazing. So I'm sure you're going to see a very good presentation, and who knows, maybe after this you also go to Michigan working with her, because that's what I did. I moved there in 2014, so she's the reason that I'm here right now working in the US. And I can tell you that being a postdoc with her, I I worked with her for 5 years or so, and I can tell you that it was very inspiring working with her. I learned a lot about how to do good research, and I also have seen her treating her students and postdocs very well and giving very good advice about not only research, but about where they wanted to go after that. So I think that this is the introduction I can do. And Lut, if you want, you can come here and give your presentation.
Well, thank you very much. This is such an honor and privilege to be invited to be the Lawler lecturer, distinguished lecturer. So I first want to thank Alice and Des, I don't know, there's Des, for starting this initiative. This is a wonderful opportunity for some of us to visit and for all of you to have this yearly gathering. So thank you for starting this lecture. I also want to thank those of you who came up with my name to suggest me. So I know Lynn was probably involved, maybe Xavi, Des himself, perhaps Mary Jo, who unfortunately is not here. So thank you very much for suggesting me and for inviting me. It really means a lot. It's a huge honor. I'm going to jump right in, and you can tell my title, Retooling Anaerobic Digestion to Maximize Waste Carbon Conversion in a Circular Bioeconomy. It's a mouthful, right? It's how we are currently thinking about anaerobic digestion technologies. And I'm going to give you a little bit of background how we got there and give you background on why we're thinking about anaerobic digestion this way. And then I'll get into some more of the technical aspects and what we're currently working on. And this slide feels a little silly to me. You can see I'm talking about refineries. And so I'm coming to Texas knowing nothing about refineries, and my first slide is about refineries, right? So, but anyway, I did it. I kept it in there. And the reason is that I want you to think about today's refineries, and this is a schematic that I took from this last year's paper from an article in Nature, so hopefully it represents things accurately. Don't know anything about refineries, at least not today's refineries. But the point is that in today's refineries, we use mostly oil as inputs, and if we look at our outputs, they're mostly transportation fuels, right? So in this paper, they talk about what the refinery of the future, 2050, perhaps could look like. And you can see that things are very different. The average size of a refinery is smaller. Perhaps that's the prediction anyway. And the inputs are very different. We use biomass and waste streams. And we have still transportation fuels, but only diesel and jet fuel. Those are fuels used for transportation that's difficult to electrify, and all the other transportation fuels are gone from the outputs. Rather, we have a large fraction of chemicals and polymers in addition to to diesel and jet fuel. So the question is, can we get there with the resources we have available? So I dug into some reports from the US Department of Energy, and this report, called the billion-ton report, talks about all the feedstocks that may be necessary to make the bioeconomy a reality. And it talks about, for example, turning all the aviation fuel into sustainable aviation fuel produced by bioprocesses. And to be able to do this, we would have to increase the sources of feedstocks. And waste streams in dark brown here in this schematic are a substantial fraction of the feedstocks that are necessary to make that happen. The billion-ton report comes from this notion that you need 1 billion dry tons of feedstocks in the not-too-distant future to make the bioeconomy a reality. And so, in addition to the current biomass streams, we need waste streams to be able to do this. We have lots of waste streams in the US, fortunately or unfortunately, depending on the perspective you're looking at. But here is an attempt to put together a list of organic waste streams that are available to us. And you can see these units are a little different than in the previous slide. Here, these are wet tons. And so million metric tons of wet waste is what these numbers represent. So we have food waste, and we have different types of food waste. We have sewage sludge. We have lots of animal manure, more than 1 billion tons of animal manure, and we have paper waste. I've tried to give you some references for those of you who want to check my numbers. This is not easy to figure out, but here are my best estimates of the waste streams that we have available. And then when you start looking at how they're currently treated or discharged or managed, you can see that there's a lot of options. I only did this for food waste and sewage sludge. But the message of this slide is that there's a lot of untapped potential. A lot of this waste ends up in landfills. And even though some people claim that we're trying to recover resources from landfills, we're not really doing it. Yes, there is biogas produced, but it's not captured very effectively. And there's lots of other things that are not done in the best possible way. So we have lots of resources. We have a lot of untapped potential. So to make this biorefinery of the future a reality, we're going to definitely need to add more waste streams. And I don't want to criticize this Nature paper because I don't really know all that much about this topic, but I added this arrow here to suggest that we'll need to add some additional waste streams to make this a reality. So I think we have the feedstocks, or at least we can improve things as it stands today. But do we have the technologies to convert waste carbon into the desired product so that we can make this biorefinery of the future a reality? And that's where I think more work is needed. I don't think the technologies are available at this point. They're at the research level, but they're not at the scale that we need them to be. And there's a lot more work to be done. And this is where anaerobic digestion comes in. I'm a strong fan of anaerobic digestion, but I also truly believe that anaerobic microbial communities in these bioreactors, basically anaerobic digesters, have a lot of potential to provide the foundation for this refinery of the future. And that's what I want to talk to you a little bit about today. And so what you see in this slide is a schematic of what's happening inside of an anaerobic digester. So there's a lot happening. You have complex polymers that are in these waste streams that are degraded by a whole suite of different microorganisms to finally produce methane and CO2, which is basically biogas. So 60% methane, 40% CO2 is your typical composition of biogas. We need to modify our anaerobic digesters to make that biorefinery of the future a reality, because obviously making biogas is not going to be good enough. I wanted to just, for the students in the room, tell you that you have a really awesome professor here at UT, and you just heard him, who knows a lot about anaerobic digestion. So if you have any questions after my talk that I can't answer, go to Xavi and ask him about anaerobic digestion. And you may have already heard about it from him in the classes you've taken, potentially. I also have a funny picture of Xavi that I wanted to show. This is Xavi at Michigan State University, not the University of Michigan. We're a rival, but we used to go there. Xavi used to go there all the time to collect the content of the stomach of a cow to seed the anaerobic digesters that he was running in our lab. And you can see Xavi here standing with a big glove, sticking his hand into the stomach of a cow to collect that biomass, those microorganisms, to seed our reactors, right? So I wanted to show that picture. But in addition to Xavi, there's another professor who you can talk to about anaerobic digestion. And here is Des Lawler. I didn't know this until recently, and most of you probably don't know this, but Lawler also worked on anaerobic digestion. And I'm saying sort of because I don't know if he really did, but I did look up his presentations, and here they are. So you can see they're from the 1980s, and anaerobic digestion is part of the title of these papers. So he did work on this, but he took it from the angle that you might expect, right? Like this one is focused on the effects of particle size and dewaterability. So that fits with his expertise, but he worked on anaerobic digestion. Just wanted to share that for the UT students, so you have two professors you can go and talk to about anaerobic digestion. So if this anaerobic food web looks a little too complicated, go and talk to them. So I mentioned that we have to modify this anaerobic food web because we're not producing biogas is not going to be good enough. So I wanted to talk a little bit about how we can engineer these microbial communities to produce higher value products. And CO2 is a byproduct of some of these reactions, and so that's a loss. It's a carbon loss. So we would like to convert the CO2 into valuable products. And so one way that we're considering doing this is to convert the CO2 using microorganisms into acetate, and then the acetate and some other byproducts like lactate and ethanol can be converted into medium chain fatty acids. And you see these two stop signs pop up here because we also need to stop the methanogens from doing their work when we want to produce these higher value products like medium chain fatty acids. So this is modifying the natural anaerobic microbiome, the anaerobic microbial community, quite a bit to accomplish what we want to accomplish. But that's where we need to get if we want to make that biorefinery of the future a reality with anaerobic digestion. And I prepared this slide here to show you that these medium chain fatty acids that we're working on have much higher values than methane or biogas. They're currently used for a lot of products already. You can see some of the applications of these products. They're currently made from other products in a not very sustainable way. So even if you're just going to produce these products, that's already a big win. But if you can make sustainable aviation fuel out of them, that's an even bigger win. And to show you that this is not completely far-fetched, I took this schematic from another publication. This is a 21 publication in which they showed the pathways to make sustainable aviation fuel out of these medium chain fatty acids. So you need to convert them to ketones and then to alkanes using different chemical processes, or you can think about future metabolic engineering pathways. So this is possible, and we are discussing and have written proposals with people in a couple of different national labs to get to that objective. We focus on the production of these medium chain fatty acids in some of our work, as I will show you, but these next steps are absolutely necessary, and that's where interdisciplinary collaborations come into play. So I've told you a little bit about how to change the anaerobic microbial communities work, but to be able to do this, you also need to think about reactor engineering. You have to think about bioreactor innovations to do this. And here's my little summary slide for those of you who haven't thought about process engineering for a while. In a conventional anaerobic digester, the hydraulic retention time is the same as the solids retention time because these reactors are CSTR, continuous stirred tank reactors. And so that means that for anaerobic microorganisms, which are very slow growing, which have very long doubling times, you need very big reactors because you need a long solids retention time to make them stay in your system, maybe 15, 20, even 30 days. But that also means that your hydraulic retention time is very large, and therefore your reactor is very large. So very expensive, large footprint. And so we started thinking about this a long time ago, 25 years ago, and we started working with anaerobic membrane bioreactors as a way of making the reactor smaller, going to a smaller footprint, so we can now separate the HRT and the SRT by having a membrane unit embedded in our CSDR system. And here you see a photo of a typical industrial a membrane system that you can embed within a bioreactor to filter out biomass, to separate the HRT and the SRT. So like I said, we started working on this many years ago. I'm not going all the way back, but I'm going to show you a few results from the work of some of these graduate students that worked on this. And we started with Adam Smith here, who was working on municipal wastewater treatment with anaerobic membrane bioreactors. And I'll tell you the story of how we were initially very excited about this work, but then we decided to switch gears and again reinvent our bioreactors and convert them to anaerobic dynamic membrane bioreactors. And that is work that we're still currently working on. So here's the beginning of that story. So Adam operated anaerobic membrane bioreactors in the lab, and he had a bioreactor in which he had 3 submerged membrane units. So each one of these membrane units here had independent sparging control for fouling control. So he had a low fouling membrane with high intensity sparging, and a medium fouling and a high fouling membrane for which there was little sparging. And so we have 3 different permeate streams for each of these membrane units. And I have some photos where you can see in this clean system, you can see the membrane unit within the glass bioreactor, and here you have a fouled membrane. I'm not going to show you tons of results, but I wanted to show you this result from Adam's work, in which he looked at the activity of methanogens in the suspended biomass of the bioreactor in green here, and then in samples that he took from these 3 different membrane units. So we have the low fouling membrane unit, the medium one, and the high fouling membrane unit. And on the y-axis, you have your relative expression of one of the methanogen's genes, the MCR8 gene. So it gives you an idea of the relative activity of the methanogens in these different samples. And what you can see is that there was a much higher activity in the biofilm on this high fouling membrane than in the other membranes and then in the suspended biomass. And we initially thought, oh, that's really cool. We can get really good activity of methanogens, of microbes on these biofilms, so that gives us additional treatment, which is true. But the disadvantage is that the methanogens produce methane right at the interface of where the permeate, the treated effluent from this reactor is produced. So you have dissolved methane in your permeate. And in many cases, we actually had oversaturation of methane in our permeate. That's a huge disadvantage because now you have a permeate that has high concentrations of dissolved methane. They're going to be released to the atmosphere. So that has greenhouse gas impacts. And that's one of the big disadvantages of this system. So we did environmental impact analysis, a life cycle analysis in this study, and this was another PhD student who collaborated with Adam on this. And in this plot, I'm showing in the y-axis the global warming potential, and in the x-axis the energy consumption. And so if you are in the green area for both the global warming potential or carbon offsetting and energy recovery, you're in a good space. We did this study in great detail, published this in ESNT a number of years ago. We're still looking at this paper all the time because it was a good study to help us think about wastewater treatment using these anaerobic MBRs. And we compared it to conventional systems. And here are some results from that initial analysis where we used your typical municipal wastewater treatment systems. And we found that the high rate activated sludge with anaerobic digestion, kind of the most conventional system you can come up with, was actually the best in terms of environmental impact for these 2 criteria. And we were hoping that our new anaerobic MBR system was going to be better than this, right? That was why we had started working on this. It turns out it was worse than anything else. And we have a really large cloud here because there's a lot of uncertainty associated with this novel technology. So this Monte Carlo simulation, so that's why we have this big, large cloud. But you can see none of our points are in the green area, so not good. And then we started thinking about what in the future can we improve this. And so we ran some scenarios with future anaerobic MBRs. And this is looking a little bit better, but still not where we needed it to be. So we had identified an exciting technology from a research perspective, but wasn't particularly promising based on this life cycle analysis. And so in the meantime, a lot of other people are doing research and even startups, and there were a lot of patents being filed on anaerobic MBRs. And one of our PhD students, Caroline van Steenbedam, got really interested in looking at patents. She found 83 patents in this time period focused on anaerobic MBRs. And she organized them in this table here where she looked at the targets that they were trying to accomplish and the strategies that they were using to accomplish those targets. And she characterized them and looked at the details of these patents. And when you see large bubbles, that means people were paying attention in many patents to these targets and use these different strategies. Or you see small bubbles, that was not a big focus of a lot of these patents. And so we were interested in seeing these things that we had identified, were they going to be popping up? Here's the overview. So you can see there's quite a diversity of targets and strategies used in all these different patents. The things that we wanted to focus on: decrease energy consumption, only 11% of the patents focused on that. And this issue of eliminating the dissolved methane in the permeate, only 2% of the patents focused on this. And that gave us additional confidence that perhaps this was not a strategy, these anaerobic MBRs, that we wanted to continue working with, and it gave us an incentive to try to reinvent the anaerobic MBRs. And that's where the anaerobic DMBRs came into the picture. So we redesigned these systems to address these problems that we had identified. And we developed this recirculating anaerobic dynamic membrane bioreactor. And what is this and how is it different? It's basically, instead of using your conventional polymeric or ceramic membrane that people use in membrane systems in the water field, we use a mesh with a large pore size, maybe 40 to 100 micrometers in size. These pores are large compared to the pores in a microfiltration membrane, for example. And you can get biofilms developed on these meshes when you use them in a filtration setup. And then once you have a biofilm developed, you can really filter out solids. So it's a great filtration device, good solids-liquid separation, but you have much lower energy demand and higher fluxes than you would have in a typical membrane bioreactor system. And by recirculating the permeate, we were substantially reducing the dissolved methane concentration because we were eliminating that oversaturation. So we were reaching equilibrium by recirculating, and so we were addressing several of our problems from before. And this is the system that we started doing research with. So here are some renderings of these dynamic membranes. We have them sometimes on a flat sheet, a flat mesh, or sometimes we have them in a tree-like structure to increase the surface area of the dynamic membrane, and then we embed them in bioreactor systems. And so this is an example of two different types of dynamic membrane bioreactors. The second one also has that recirculation that I talked about. So then we started shifting our research to focus on using these anaerobic dynamic membrane bioreactors for different applications. And so you see Xavi's picture here because he used one of these to study the food waste treatment using this system, and he was focusing on biogas production at the time. And then we later on used them for co-digestion of food waste and sewage sludge, and we also started working on producing renewable natural gas instead of just biogas. And I'll show you a couple of examples. So this is work where we used this system that I showed earlier. We call it our two-phase anaerobic dynamic membrane bioreactor system. And that first phase simulates the stomach of a cow, the rumen. And you can see some of these conditions here with a solids retention time of 4 days and a really short hydraulic retention time, so a very small footprint. And the second reactor is a methanogenic system, which also has a small footprint because the hydraulic retention time is much lower than your typical system. In our co-digestion work, we continue to work with the system, but now in addition to food waste, we also added sewage sludge, and I'll show you some results where we obtained really high hydrolysis rates. So we calculated the hydrolysis rate constant, which I'm plotting here in the y-axis, for this first phase anaerobic dynamic membrane bioreactor. And then when we we compared this to studies in the literature that also looked at hydrolysis, you can see that we got at least double in terms of this hydrolysis rate constant in this particular system. And the big thing is that we have a much smaller footprint than these systems, which have lower hydrolysis rates. And when we add sewage sludge, this was the food waste data. If we add sewage sludge, we get even better hydrolysis rate constant. So there is some synergistic effect of doing the co-digestion that we haven't fully understood why that is, but these are observations that we've made. In the second phase, we produce biogas, mixture of methane and CO2, and we get good methane yields, which I'm plotting here in the y-axis. And these yields of methane are comparable to the yields that we observe in an anaerobic membrane bioreactor, but we have mitigated several of these disadvantages that I talked about before. So we have a comparable system in terms of methane yield, but we've addressed a lot of the disadvantages. We also did an elaborate environmental impact analysis of this two-phase anaerobic dynamic membrane bioreactor system. And again, those of you who are interested, go and check out this publication. I'm just showing you a big overview table here. We compared this technology here, this two-phase anaerobic dynamic membrane bioreactor system, with lots of other strategies that are out there to manage these different waste streams. And you can see in my overview, qualitative overview here, is that our system came out at the top in terms of the environmental impacts for these different categories. It's interesting to look at when you see the system that looks best, that looks second best, is your conventional co-digestion system in a traditional anaerobic digester. So, but our system was considerably better for all of the impact factors, for all of the parameters that we evaluated. In a current project, we are collaborating with others to upgrade our biogas to renewable natural gas, and I just wanted to briefly touch on this, give you a flavor for these collaborations that develop when you work with people with different areas of expertise. We worked, for example, with Argonne National Lab. They developed a new electrochemical system to upgrade the biogas to pure methane using electrochemistry, but then at the same time, they produce hydrogen using electricity. I mean, they like to say renewable electricity because that's the goal. And we bring bicarbonate into solution, and we have the hydrogen that's produced by the electrochemical cell, and we produce them in the ratios that are necessary for hydrogen utilizing methanogens to convert the CO2 that otherwise would be a loss into more methane. So now we produce renewable natural gas close to 100% methane that can be fed directly to the natural gas grid. And that's happening as we speak already around the country, not using this technology, but using membrane separation, right? So the infrastructure is there to produce renewable natural gas out of biogas, but we would just like to have some technologies that make that more sustainable. This system here that converts the bicarbonate and the hydrogen to methane is also really interesting. It's a membrane biofilm reactor system. but that is operated at Northwestern University, another collaborator on that project. The group of George Wells, some of you probably know George. So that's how we've continued this work on the anaerobic dynamic membrane bioreactors. And I alluded to the medium chain fatty acids that we want to get to, right? So I wanted to tell you a little bit about the projects in our lab that have focused on MCFA, or medium chain fatty acid production. This work started with Shilva Shrestha, who is a former PhD student in the lab, and has continued with these two PhD students. Diana Kidd actually recently finished her PhD as well, but we have a new student, Jinran Zhang, continuing this work. In this work, you need to use different feedstocks. You need to combine electron donor compounds like ethanol or lactate with short-chain fatty acids to elongate those short-chain fatty acids to medium-chain fatty acids. And this is a biological process that can take place naturally, so people have started doing this in your typical CSTR reactors. The problem now is that you have to extract these medium-chain fatty acids because they're quite toxic to the microorganisms. And to make this process feasible, people typically have used multiple filters to be able to remove the suspended biomass and other suspended material to be able to send this filtered effluent into extraction units. And here's a schematic of a typical protraction unit. And that gives you your pure MCFAs. And so this is a pretty complex system, difficult to scale up. So we thought, what if we use our dynamic membrane bioreactors? We hopefully could skip these filters, these multiple microfiltration steps. And that's exactly what Shilva evaluated. She did a lot more, but I'm just showing you some results from how she first was running a system without dynamic membranes. And so she had a relatively high level of suspended solids in the permeate of her bioreactor. And then as soon as she turned on the dynamic membrane system, she got relatively low suspended solids. And in her bulk liquid of the bioreactor, she kept increasing biomass. So she had an increase in suspended solids concentration. We have recently, with the work of Diana, moved to using anaerobic dynamic membrane bioreactors directly combined with some novel extraction systems. And this is a collaboration with people in the Chemical Engineering and Material Science Department at the University of Michigan. They've developed this system that they call CLEANS, continuous liquid-liquid extraction and in situ membrane separation, that they believe is much beneficial compared to conventional extraction systems like the protraction system. And so we've integrated their system with our anaerobic dynamic membrane bioreactor to directly extract those medium chain fatty acids. And Diana, instead of using ethanol as the electron donor, Shilva used beer waste with a high level of ethanol as her electron donor source. Diana used acid whey, a waste product from the dairy industry that has a high lactate concentration as her electron donor. And then she combined that with short-chain fatty acids. And she ran this bioreactor for a long time period. So you can see here in the x-axis. And she tried this at different organic loading rates. She used initially simulated acid whey, and then we switched to an actual acid whey that she got from a dairy. And when you look at results, first the suspended solids concentration, you can see again that the permeate concentration of the suspended solids was low, so the dynamic membrane was performing as expected. The influent is what this darker pink line represents. The influent suspended solids concentration made this big jump here because we switched to real acid waste, which has a much higher suspended solids concentration. And then her biomass concentration in the bulk liquid is represented by this line here. But you can see the dynamic membrane is performing really well. So she was able to send that permeate directly into that novel extraction system. And she got really good yields of these medium chain fatty acids. Here we're plotting their concentration in millimoles per liter as carbon for the different compounds that she measured. And the ones that we're most interested in are caproate, heptanoate, and octanoate, the C6, the C7, and the C8 medium chain fatty acids. And you can see, for example, the C6, the caproate, popping up here as one of the dominant products in her reactor system. You can also see that there was a decrease in the concentration after she switched to the real acid way. And this is, we think, due to two reasons. One is that she used a lower loading rate, but we also think that there may have been some inhibition effects that started to pop up in her system. So toxicity of these medium chain fatty acids, we're using these extraction units exactly for that reason, to remove them as soon as they're formed, but we still build them up to decently high levels that we expect that there is toxicity. There's not a lot of research done on these toxicity effects in these mixed community systems, so we don't have a good handle on this. But we looked at some pure culture studies. So this is a plot from a pure culture study of two different organisms that can produce these medium chain fatty acids. And we looked at, or this study looked at growth rate inhibition in the y-axis here, and then the concentrations of the different acids added. And when we look at caproate, that C6 compound that we were interested in, you can see that you get complete inhibition at a concentration of maybe 30 or so millimoles per liter of that compound. And our compounds, our units were a little different. Remember, they were in millimoles as carbon. So if you convert them, we were having the exact same concentrations when we were at that highest level. And I can, for example, show you a different rendering of the same data. So you can see here at their highest point, we were at caproate concentration of about 180 millimoles per liter as carbon, which divided by 6 corresponds to that threshold that we saw in that pure culture study of toxicity, complete inhibition. So we, of course, don't know what this means for our system, so we are trying to dive further into this by studying the microbiology. Diana focused in on one period in the operation of her system when she had these really high levels of these MCFAs. And then she also looked at the microbial community, and she identified a number of presumptive chain elongators, microorganisms that can produce these MCFAs. And she, for example, in this plot, shows the total relative activity of these 3 presumptive chain elongators. And you can see there's quite a bit of up and down, up and down. We were not able to make complete sense out of this. We had hoped that we could see some indication of maybe the activity would be crashing, but it doesn't seem to be the case. When you look at these populations individual, this is again the relative activity of these 3 populations of presumptive chain elongators. You can see that their relative activity changes over time, and during this last period, one of these popped up as the dominant active population. So we're hypothesizing that we need to get a better handle on which microbes, what's the relative toxicity threshold for these different microorganisms, and can we perhaps engineer these microbial communities to deal with these toxicity effects so that we can get higher yields of these MCFAs without having the system crash on us, basically. And that's where we're going with this work. And I wanted to show you this slide. It's kind of a schematic of a thought process that we've been working on in collaboration with Chris Lawson from the University of Toronto and Bradley Biggs, who's in chemical engineering at the University of Michigan. And it's thinking about using this work that we have been doing, which we tend to call the top-down microbiome engineering approach, and combining this with their interest in a bottom-up microbiome engineering approach to try to overcome issues like product toxicity, as I discussed, or improve our yield challenges that we still have. And so this is a direction that we think we'll be moving into with this work on MCFA production, but we want to continue to do this in these dynamic membrane bioreactors because we think they have real potential to make something that can be scaled up in the not too distant future, so that we can develop real technologies that can be implemented in the biorefinery of the future by 2050 is, I think, what that paper indicated, right? So I'm going to conclude by just kind of recapping some of these points that I made. We need to focus on technology development if we want to make that biorefinery of the future a reality, especially if we're going to use alternative feedstocks like these organic waste streams that I talked about. Anaerobic microbiomes have great potential to accomplish this. And we think these dynamic membrane bioreactors are necessary, or at least an alternative technology that allows you to have a small footprint with low energy cost. We can't just stop there because nothing is ever easy. We need to integrate this with other technologies, or you need to work with chemists or metabolic engineers to think about upgrading to higher value products, right? So we're not down just with our bioreactor work and with our anaerobic microbial communities. We need to collaborate to work on integration with other technologies. And then finally, I do think that this combination of the top-down and bottom-up engineering microbiome approach is one that has a lot of potential to overcome problems, and I have focused in on product toxicity and yield challenges. So I really want to conclude by thanking my funding agencies and collaborators, and I've already alluded to some of the collaborators, and I referred to a lot of the students, but I want to call out one collaborator who's been with me on many of these projects, Steve Skurlos. He's a professor at the University of Michigan, and I started working with right when I moved to Michigan. He is in mechanical engineering and also has a joint appointment in my department, Civil and Environmental Engineering. The students who I highlighted throughout my talk, and some of them were postdocs and so on, but they, of course, deserve most of the credit. They did all the work. But I don't want to forget some of the undergraduates and master's students and visiting students, and you see many of them because this work is really demanding, and all of these PhD students and postdocs want to have at least 2 of these more junior researchers work with them. And so we have a large research group thanks to all these people who contribute to our work. I actually have another photo which shows my current research group, and there's a lot of the undergrads that I didn't include in that previous plot included in this picture. So I will end here. I have my conclusions up to trigger your memory in case you have any questions that you would like to ask. Thank you very much for your attention. There's a microphone here too. Yeah. There's one there.
All right. So great talk, Lut. It was fun to see it. I've seen Shilva present before on the dynamic membrane bioreactor. So I work with membranes, so I think about this a lot. And I've asked this to her, and I've thought about this many times. Why does it work better? It just has bigger pores. It has the same kind of biofilm, maybe. Why does it work better?
Well, I mean, better, what do you mean by better, right? So it doesn't work as well with suspended solids removal. It's not as good of a filter as a traditional membrane, but it allows you to have much higher flux.
Is that it, though? But there's other things that you're seeing, right?
We see a lot of biological activity in the dynamic membrane as well. And in a traditional membrane, sometimes you have a little bit of biological activity on the biofilm that develops in your fouling layer, but it's not as substantial. Here, we're actively trying to promote that biological membrane, so if you will. We call it dynamic because we're doing some fouling mitigation, and depending on the system, sometimes we don't do much at all. In some systems, we break up the membrane quite regularly, and so it's removed and it forms again very quickly. And so it's a dynamic system. That's where that term comes from. Yeah, so it's completely different concept than a conventional membrane in many ways.
Yeah. Makes sense. Yeah, thank you.
Hi, thank you for the great presentation. My name is Eric. I was just wondering, for your extended duration simulations, did you have to add any supplemental alkalinity to maintain the pH in your system, or is it pretty stable throughout the...
Yes, we actually did supplement sodium hydroxide in our system because the system naturally goes to a lower pH, and we were trying to control the pH at 5.5. And so if you don't use a base, you will push it down further. Yes. So that's a disadvantage of the system. Yeah, it's expensive.
So, Lud, I'm wondering what happens with the solids that you have to waste from the reactor. What are we going to do with them? Because I mean, it's a pretty nasty waste stream when you think about it, because it's got all sorts of volatile.
Is it? I mean, it's just a biological sludge. We have much less of it than in your traditional system. We maintain these really high solids retention times, so we don't need to waste very much. But yes, you do need to waste at some point, and then you just have a smaller amount of the same thing. So
that would be like material we send off to the landfills and is largely inert.
Well, it's stabilized, is what we call it, right? So it has gone through extensive stabilization. So we tend to currently land apply it or put it in a landfill or incinerate it, or Chavi is trying some new technologies to get more energy out of this sludge. So there's a number of different things people are doing or researching. But yes, you can't get rid of it completely. Yeah. But it's not more nasty than anything else, by the way. I think it's less nasty because it's so stabilized.
I love how wastewater people talk about nasty and levels. Have you thought about, you talked about food waste, have you thought about grease traps and others where it's pretty concentrated? I've seen, I think one of Dr. Molina's last PhD students did some blending of grease traps. So it's a pretty concentrated source into the digesters. And kind of what are your thoughts on that?
Yeah, it's actually an amazing feedstock that's very difficult to manage. So if you could, we're actually in a different project. We're trying to model these anaerobic digesters. We're collaborating with a food waste digestion system, and we're trying to help them decide how to blend their different food streams and how to blend in, for example, that really energy-rich stream. The problem is that you can very quickly get an unstable digester if you add in too much, because you release these long-chain fatty acids, and they're quite toxic, and there's inhibition effects and so on. But if you blend them in at the right ratio, you get a huge benefit, and you get higher biogas yields and higher RNG. Yeah.
Lute, this is Mike Cavanaugh. How are you, Lute?
Hi.
That was a great talk, and it seems like you're doing some incredible work there, and it's taking a long time to do all of these tests. I'm wondering if you've got a theoretical foundation that you're able to play with in terms of kinetic reactions, the old Michaelis-Menten equations. Yeah. Like we used those for many years in the activated sludge world, and it's been very effective in terms of defining where the limits are in your... Is there some theoretical foundation that you're working on in that, along with this theoretical, the experimental work?
I alluded to this in my answer to Carrie's question, because we have, like the activated sludge people, we have these bioprocess models that are built on monokinetics, Michaelis-Menten kinetics. So we have the anaerobic digestion model, which has incorporated all these different populations And you can use that tool to help you predict either steady state or dynamic performance of these systems. It's quite a complex model, as you can imagine, because it's a complex microbial community, quite a bit more complex than your activated sludge models. But it's a great tool, and we're trying to use this in practice to help predict stability, for example, of these digesters and how to decide how to blend in food waste. We haven't used it as much in these research projects, and so I think that's a great suggestion to try to do more of that theoretical modeling and predictions with these research projects. These models are quite complex to run, so I only have a few people in the research team who are proficient with these models, so I just have to kind of make sure everybody becomes familiar with how to run them, and then we can use them more effectively. That's a great suggestion.
Oh, it's on. Okay, everyone can hear me. Hi, I'm Ben, student of Dr. Katz. I don't really know much about biotreatment. I work more with PFAS, but I was interested in the bioreactor's capability to remove things that, like beyond the fatty acids.
Okay, so for example, PFAS? Is that?
Yeah, any
yes.
contaminant.
Yes. So yeah, I mean, that's a really good question. I think one of the biggest constraints in a lot of this using waste streams is the fact that, if you use food waste or if you use sewage sludge as your source, you have PFAS, you have microplastics, you have other organic contaminants that are not all removed. They're removed to some extent in these bioreactor systems, but they're not all removed. So those might end up in that nasty sewage sludge that we talked about, and that's becoming a bigger problem because now we're finally actually paying attention to this. So we are not addressing this. We're thinking about this, and we have some ideas for how to deal with this, but none of these projects I talked about have addressed these contaminants. Xavi, again, was telling me earlier today he's working on measuring PFAS in some of these technologies that he's developing. So definitely some people here at UT are thinking about this as well. So yeah, I don't want to underplay your question. It's a really important one, and it might be in the end a driver for a lot of this work, because if we generate these waste streams that we have to do something with, right? And if we cannot put them in a landfill or we cannot put them land apply them, we cannot treat them properly, and there's going to be regulations. What are we going to do? Yeah.
Yes, I was just curious. Have you measured the nutrient limits coming out of the liquid waste stream? And I assume the liquid waste stream goes back to basically the head of a treatment plant, or what's the thought on that?
Sorry, you'll have to repeat your question. I'm not following exactly.
Have you measured like the phosphorus and the nitrates and the other nutrients, what we consider nutrients, coming out of the waste stream?
For different projects we have. For most of the ones that I talked about, this is not a concern. The blend of food waste and sewage sludge, for example, gives you sufficient nutrients, but you have relatively low ammonia concentrations and phosphate concentrations, so there's no typical toxicity effect. If you're concerned about having residual nutrients, that is something definitely you have to pay attention to if you want to discharge or you want to reuse water. So yes, nutrients are not removed in these technologies, and you'll have to add additional technologies if needed. Yeah. In some of our other projects, we're working on nutrients removal, but I didn't talk about that today. Yeah.
Lut, my mind keeps going back to the 500-day study with the results there. First of all, it's kind of amazing. But the productivity, if I understood it, goes up and up and up and up, and then it reaches some peak, and then it goes down, 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 down.
Yeah.
So I'm wondering what was at that peak that was different before and after, and could that be understood well enough to be able to kind of say that's where we need to operate, and Can we operate that on a steady state basis?
Well, yeah, I probably should admit the study wasn't designed all that well. Like we're still, trying to figure things out, and that's partly why it took so long, because there's lots of things you do with these bioreactors. We felt from the beginning we wanted to work with real waste streams at some point. And so we got this really good performance with this synthetic dairy waste. And then we decided, okay, we got good performance, it's time to switch to a real waste. And that's when things crashed. Is it because we switched to the real waste? Was there other factors like toxicity that I think is a factor, but is that the only factor? So it's pretty much a black box at this point. We're trying to figure it out a little bit, and I alluded to some of it, but we haven't made a ton of progress on that. So I don't have a good answer for you. Yeah.
Hi, I'm Carlo with Kirsted's group. I had 2 questions. One is about the food waste, how the composition of it affects the efficiency, but that might not be feasible because it adds a lot of complexity maybe to try and get different food waste types. And also the other question was the seeding. It must be very interesting to put your hand inside of a cow, but that is a great seeding source. And I guess I'm wondering how much does the seeding source affect? Is it the initial seeding dies and then it's just up to the new anaerobes that take over? And what other anaerobic seeding sources are possible?
Yes. Yeah, the more I work in this field, the more important I think the seeding source is. So I used to think, oh, it doesn't matter that much. Let's take a few anaerobic biomass sources and blend them, and things will turn out okay. But I think there's more and more studies that suggest that the seeding is important. And so going to, for example, that the stomach of the cow environment, rumen environment, it does give you organisms that may not be in any anaerobic environment that are really helpful with some of these processes. So inoculum source is important. Once you get your system going, we often store inocula in the cold room in the fridge, and they are there for months, and then we want to start another reactor, and they keep going again. So we don't always have to get fresh inoculum from a cow. But there are plenty of other anaerobic digester sources out there. We often just go to our local wastewater treatment plant and get a sample from the anaerobic digester, depending on the study that we need to do. In terms of your question about food waste, for a lot of our work, we just worked with cafeteria waste from the university, so post-consumer waste, but we have tried a few different things, pre-consumer waste and so on. We haven't, some people have done putting together a synthetic food waste that simulate, certain things. We haven't gone that far to properly characterize it. I mean, we characterize it to the extent that's necessary to know what is going into our system, but we haven't really worked on the feedstock components. But in that project that I alluded to, with Carrie's question, we're trying to help a real full-scale food waste digester to help think about how to put their daily feedstock together, because they get food waste from all these different sources, and they have an opportunity to really blend it in a way to get the highest possible RNG yield. They're actually converting their biogas into RNG and adding it to the real grid. So there's a real opportunity for gain there. In addition to this, maybe you haven't thought about this, but they get this food waste from all these different sources that are now not putting it in a landfill. So they get tipping fees. And so their revenue is not just coming from the RNG, but the revenue is also coming from the tipping fees. And so sometimes they can get a lot more tipping fees from accepting some food waste that is likely going to make their digester unstable. And so they have these questions: should we accept this or not? What are we going to do? And so this is where our modeling can come in and help them to make decisions. Yeah.
I think this
I'm first? Okay. My name is Matt. I'm a program alumni. So, as you're describing all this, I'm thinking about how does it relate to what is happening in the anaerobic digestion industry, municipal industry now. And right now, a lot of people are doing things like thermal hydrolysis, and there's a lot of talk about even microbial hydrolysis, things like that. And you also spoke about the impact of the material coming into the system. Have you guys looked at all at how things like perhaps thermal hydrolysis might affect the operation of your system if you maybe are feeding it at a higher concentration, or it can impact the viscosity, which might affect membrane performance? Has that been looked at all, or is that even something you think would be worth looking at?
Yeah, I mean, different people have done lots of different work in this hydrolysis space. The way we have tackled this is to focus on biological approaches to improve hydrolysis. And this is where this idea of using the rumen bioreactor, so biomimicry, if you will, of a very good hydrolysis system that's in nature. So we have used that approach. And that's been very productive for us because it's relatively simple. It's just another bioreactor that you run at maybe strange conditions, but it's just another bioreactor. Whereas a lot of these other hydrolysis approaches, are higher temperature or higher pressure. And so we've kind of stayed away from that in our research. I'm not saying that in full-scale systems that none of those have a role. Clearly, some people are using them, some of them. But that's how we've tackled it. Yeah.
Lou, one last question for you. I don't know if you're familiar with the serum lab that Geosyntec developed, but what the serum lab developed was the growth of anaerobic microorganisms that could be used to bioaugment in situ remediation technologies. Maybe there's some option here for growing microbes that are appropriate for producing these medium chain fatty acids.
Yes. Yeah,
Because
actually,
that was 15 years ago.
Yep.
It was really innovative work, and maybe there's some direction here that you could think about for that too.
Right. My last slide kind of alluded to that idea, where we would basically, come up with microorganisms that can deal with these toxicity, can produce compounds at higher yields, and perhaps construct our own simplified microbial community. So yeah, we're thinking in that direction, but we haven't done too much in it yet.
That's great. It's been successful with anaerobic digestion for anaerobic degradation of benzene and other chemicals, so you're probably familiar with that.
Yes. Well, Elizabeth Edwards' work, maybe, and
she was the founder of that.
Yes. Yeah. That is amazing work, but I agree with you. We should think more in that direction. Yeah.
Lute, another round of applause. Really thank you for your There is a reception outside the door, so please go help yourselves. And as we said, part of this is community, so this is the community sharing portion, so please take the time to get to talk to each other.
So I have a request for the UT faculty. I know Des would really like to get a photo. So can we come maybe to the front of the room and take a photo before everybody goes to the reception? Is that okay, Des? Yeah.